Okay. Just gonna adjust this. How low does this go? Okay, that's fine. Hello and welcome to the day. I believe we've only done 3 of these so far. So this is technically only day 3 of the revision streams. What we're doing today, I need to center that. So for those unaware, Streamlabs is a bit stupid and it flipped the orientation. So now when I write something, if I have it in the angle that I used to have it, it is now inverted, which is so stupid. What it basically means is I can't see chat now. In order for me to see chat, I have to go all the way behind, which is stupid. It's stupid. It makes no sense. But anyways, today we're doing some biology revision once again. I was gonna do some history, but again, I prefer to stick with biology revision for streams because it's more universally, I guess, understood. So let's do some biology revision for A-level, AQA, and we're gonna just look at some practice questions. We're gonna go to the AS level topic for cells. We're gonna do, and then we'll jump to biological molecules in a little bit. So we're gonna start off with cell structure and microscopy. Then we'll go on to the cell cycle and mitosis, and that's all we're gonna be able to do because that's all the topics we've done. All right. So, it's Latin, is a single-celled eukaryotic organism. The diagram below is a photograph of you, taken through an optical microscope. Explain why it's not possible to determine the identity of a structure labeled X using an optical microscope. Well, clearly the resolution is low and the magnification, I guess that's it really, just the low resolution. It's an optical microscope. Obviously those organelles there, the structures, are smaller than the wavelength of light, which is why they appear so blurry. Yeah, resolution too low, the wavelength of length is too long. Exactly what I said. I did just cheat there. I just looked at the answer by accident to the next one. We'll try and logically work it out though, even though I know the answer. So these cells ingest bacteria and digest them in the cytoplasm. So I'm assuming that's a phagocyte. Describe the role of one named organelle in digesting these bacteria. So we have to describe the role for 3 marks. Alright, so actually no, I can still answer this one. So that's obviously lysosomes. They hydrolyze material. So hydrolysis, using a water molecule to break down. I don't exactly, again, you guys have to remember, I've just done a week of NCS. I could not be I could not be bothered at all to do anything today. So I'm very tired, very shattered. I can't remember the basic definitions of words. So please bear with me here if this seems obvious to you, because it'll be obvious to me in about a day, but they hydrolyze the material, I guess, outside of a lysosome. They break down worn out cells, but it says in digesting bacteria, so I guess it's going to hydrolyze, I guess I'll just put cell matter. I'm sure there's a better word for that. How am I going to get 3 points out of that? How am I going to get 3 points out of this? I guess we could specify that it like breaks down. It breaks it down. I can't think of more points for this. I know lysosomes obviously are responsible, I guess, for like cell death. Like basically destroying a cell when it's close to death, and it replaces, or I guess, breaks down worn out organelles. How would that apply to bacteria though? I've kind of specified the main point there, that it hydrolyzes cell matter. Okay, let's have a look then. Let's see if I can remember some stuff. Ah, I did not know that. Okay. So yeah, I would have literally just gotten a mark for saying lysosomes. In fact, I'll get like a green pen out here so I can mark it. There we go. Hydrolyzed cell matter. So what you would actually have had to say there is hydrolytic, hydrolytic, guys, I'm tired, sorry, hydrolytic enzymes, it releases them. Okay, so we were never told that, so I'm assuming that's something we'd learn in the infection and response topic, maybe. So they hydrolyze by literally releasing hydrolytic enzymes. I did not know that. So there we go, something new. So they don't just hydrolyze it, you have to be more specific and say they release hydrolytic enzymes, which I assume, they break down the bacteria. Fuse with vesicle. I'm not too sure what that refers to. You know what I'm going to do? I'm going to go pull up my biology revision document. See if I've already wrote down some of this stuff. And I'm going to go quickly check chat to make sure no one said anything. Good. Okay, biology notes. Let's see if I've wrote down any of this. Hydrolyzes material that's ingested by phagocytes. Here we go. Now my memory's getting up to me. Exocytosis. They release the hydrolytic enzyme outside of the cell to destroy material around it. Okay, so they perform exocytosis. So I'm assuming the enzyme is then put into a vesicle and it's transported outside of the cell, where it then, this hydrolytic enzyme breaks it down. That's an interesting one. I think the Mac scheme was a bit vague there, so I couldn't really figure out what I needed to say. But yes, okay. So lysosomes do hydrolysis to material digested by phagocytes, and in order to do that, they release these hydrolytic enzymes, which I guess happens because of exocytosis. Interesting. Okay. See, my memory's jogging back a little. So I probably would have gotten like 3 marks so far out of 5, maybe 2. Calculate the actual length of the cell. Alright, we can do that. I'm not going to actually give you the answer because I don't have a calculator with me. I do, that's a lie. I do have a calculator with me, so we can answer this one. Calculate the actual length. Oh, we need a ruler. Well, shit. I can't do this because it's literally paper. Okay, if I look at the mark scheme, I might be able to figure it out. And yeah, it just does not show the calculation. Alright, great. I would have done it if this was on paper. Give 2 structures found in all prokaryotic cells and eukaryotic cells. This feels like it's going to be a bait question, where it's going to be something really obvious and I'm going to overthink it. I'm going to be a prick here and I'm going to say membranes and cytoplasm, but I feel like that's bait. I feel like it's going to be bait. Cytoplasm and membrane. Yeah, okay. You could also just say ribosomes and DNA. Which, yeah, now I'm thinking about it, yeah, that would have been a very obvious one to put. See, I said it very riskily there. Obviously, it's true, they all have cell membranes and they all have cytoplasm, but that was a bit of a silly thing. I should have thought a tad deeper there. And yeah, they of course all contain DNA. All prokaryotic cells contain circular DNA plasmids. Scientists have found that the rate of plasmid replication is faster in cells growing in a culture with a high concentration of amino acids. So just one explanation for the faster rate of plasmid replication in cells, I do not know. Have we done anything about how protein... Okay, my thought process here... Amino acids obviously form together polypeptides, proteins. So surely more amino acids means more polypeptide chains, more proteins. No, but I've just robloxed myself there. Plasmids. I honestly don't even recall how... I do not recall anything that we've learned about this. Scientists have found that the rate of plasmid replication is faster in cells growing in a culture with amino acids. Again, I can't think of anything that we've learned about this. This might be a topic that we're going to do with our teacher soon, because we're doing a protein-related thing. No, we're not. We're past proteins. We're doing enzymes now. Have we done this, or have we not? Okay, well, I would just want to put about amino acids. Let's see what... Oh! Bruh! I kind of... Okay. Yes, I would have maybe gotten some of this. Amino acids are used in protein synthesis. So, more enzymes, because obviously enzymes are proteins, for plasmid replication. I didn't really know that. We have not done this. I did not know that. I mean, I get amino acids obviously formed together to make proteins. Yeah, that's a basic fact. But I was not aware that then these proteins obviously... I know proteins, enzymes are just proteins, but I was not aware that... enzymes were used for directly replicating plasmids. That was something new to me there. Actually, hang on, I've recalled this word. DNA polymerase. That's obviously the enzyme. More DNA polymerase. Amino acids are used in respiration. I feel like I remember that. More energy for the... Okay. So I could have gone for two paths there. So, amino acids are used in respiration. How? How are amino acids used in respiration? I can't recall this. I cannot recall. Amino groups are yeah, we have not done this. What the fuck? Converted into urea and excreted by urine. We've not done any of this. I don't really think I would have gotten any marks for that, but I feel like we have not done this. I feel like I'm doing stuff that we haven't ever covered here. Oh, hang on, no, centrifugation. Oh, I know this. Okay, this is a nice one. What can you conclude with the figure above about a structural difference between the plasmids and the circular DNA? Explain your answer. What can you conclude from the figure above about a structural difference? Plasmids are more dense, which is why they float up to the top. Circular DNA is less dense, so that's why they're closer to the bottom. That's it, right? Yeah, circular DNA is bigger. Wait, no, I got it the wrong way around. Oh my god, yeah, obviously the denser stuff sinks to the bottom. Batty crease. I would have gotten that. Because the band is further / lower in the tube. I feel like I wouldn't have said that. I just would have said the pellets are like the strip. The strip of plasmids is above the circular DNA. It's higher up in the liquid. I don't recall what you call it. It's some word that as soon as I say it, I'll recall. And it looks like I didn't write it down. Okay. Okay, well, I might have gotten a mark out of 2 for that. Because I said it the wrong way around. And the band moved further. The band moved further. I mean, I get that, but I feel like that's a stupid thing to put on a mark scheme. Surely the more obvious thing would be the circular DNA is further down, which means it's more dense. Anyways, describe how a sample of chloroplast could be isolated from leaves. Oh wow, how many marks? 4? Oh, this is a beautiful question. We'll write it down. So if it's 4 marks, I guess it's 4 points, but because it's A level, I'll add an extra 1. So you would have to firstly get the leaf, and I guess use a pestle and mortar to, I guess, crush it down. While crushing it down, you will then add your cold isotonic buffer solution. Then, once you've kind of mashed it down, that is your kind of homogenization. That's a word. That's your homogenization done. So then you would place it in a centrifugation. And you would let that go for a certain amount of time. I don't know whether you need to specify time, but I would just say put it in a centrifugation. I guess maybe put it in a, it's not a beaker, put it in a tube, centrifuga, and then you should get a pellet of nuclei, because it's the densest, then repeat it, and you will get another pellet of chloroplasts. I feel like I missed out something there, and it's going to hit me like a bloody train as soon as I hear it. Ready? Break open... Bruh, that would have been a mark. This whole thing would have been a mark. Break open cells / tissue and filter. Oh, filter! That was it! So you break open the cells, you homogenize it. You would then filter out the debris, yes. You would then add the cold isotonic buffer solution. In cold, same water potential, pH controlled solution. Oh no, okay, that would have been 2 marks because you would homogenize it in that, yes. Centrifuge to get your pellet of nuclei. Then I should have added that you would filter out, because obviously why would you want a pellet of nuclei and then a pellet of... Okay, that makes total sense. I understand where that was coming from. Centrifuge at a higher speed, yes. I think when I was talking, all I said was do it again. So no, I might have gotten 3, maybe 2 worst case scenario marks for that there. I got that right, I got this right, I just didn't specify that you'd have to filter out the debris. Because this is strict as well. I might have only gotten 1 mark for this whole bit because it said and filter. Hang on. No flipping way. Hang on. This is made me go, wow. Homogenize and filter. That's 1 mark. Just for literally saying homogenize and filter the leaves in the cold isotonic buffer solution, then centrifuge it and remove a nucleus, and then spin at a higher speed to get your chloroplast. Wow, so I went on a whole ramble about using a pestle and mortar. No, you literally just could have said homogenize it, filter it in a cold isotonic buffer solution, and you would have gotten half marks. Yikes, these mark schemes are getting so... This is why I hate the biology mark schemes, bro. They are so picky with what you can and can't say. And I get, obviously, it's biology. You can't just make up information. But, bro, the mark schemes on the GCSE ones were putrid. And now the mark schemes for A-level seem even more like, even more stupid. Oh, hello. I'm so sorry if you're still here. For context, I'm doing A-level biology, and I can't see chat without having to literally go all the way behind the camera, thanks to the new setup. So I'm sorry. Hello. Happy Vampire Night, man. My apologies for... I was not ignoring you. Again, everything is basically upside down if I have the camera facing me. So I have to have the camera, like, the other way around now. So I can't see chat. Oh my Christ, these questions are getting worse. Scientists grew 2 groups of plants. I feel like I can't do these questions yet, I feel like I can't do these questions because I am lacking a lot of information about some of these. Massive protein, massive chlorophyll. Give one feature of a chloroplast that allows protein to be synthesized inside the chloroplast, and describe one difference between this feature in the chloroplast and a similar. What the feck? One feature in the chloroplast that allows protein to be synthesized. 70S ribosomes. It's different because the rest of the cell has ATS. That's the answer, right? That has to be... What? That is not the answer? DNA is not associated, but nuclear... What? What is bro cracking on about? Oh, no, there, I would have gotten it right. I just didn't scroll down. Okay, I got it right. Ribosomes are smaller than cytoplasmic ribosomes. 70S ribosomes in chloroplast, ATS in cytoplasm. Okay, I would have gotten 2 marks. I just should have added the bit about cytoplasm. Yeah, guys, I cannot see chat. I can see it now. So hello. Hope you're doing well if you're still watching. Again, I can't see chat without having to turn around, and I can't even check on my computer because it just kind of lags and doesn't work. So if I'm not ignoring you guys, I swear to God I'm not. I'm just doing revision, and I keep on getting lost in the questions here. Right, well, I would have gotten what? Maybe 2 marks for that? The ratio of protein to chlorophyll in control plants is 9 to 1. Oh, don't be like some stupid ass math question. Use the... How many marks is this? This is 1 mark. Okay, if this is overly complicated, use the information to calculate the ratio of protein to chlorophyll in iron deficient. Okay. So if the mass in the control is 40 to 10, that's a 4 to 1 ratio. Not a 9 to 1 ratio. Why do they pull the... The ratio of protein to chlorophyll in control is 9 to 1. I'm utterly baffled. What does this mean? The mass... Okay, hang on. So maybe it means the mass is 40 for this. The mass for this one is 10. I'm baffled. I looked at the answer. I don't know how you would get it. All it literally says is 36 to 1. The only way I can think you would do that is if you take off the zeros and then just multiply. But I'm kind of not satisfied with that answer because I don't know how you would have gotten it. I guess you're multiplying the mass by the ratio. I'm baffled here. I'm so confused. What? The ratio of protein to chlorophyll in control plants is 9 to 1. So that would be 90 grams of, let's just say 90 milligrams of protein per 10 grams of chlorophyll. So this one, it's 40 to 10. So 80 to 20. But they're both on about the control variables. I have no idea. But that question's... How is a 1 mark math question stump me? This one makes 0 sense. Am I missing a table or something? Hang on. Maybe percentage of control 40%. So there's only 40% of protein. But that still wouldn't give you... Nah. How is a 1 mark math question with the vaguest answer in a mark scheme? How does that stump me? I need to Google the answer. This is going to annoy the hell out of me for the rest of the day. Google, please. Okay, thank you, Reddit. Yeah, someone's asking the exact same thing. Aren't both ratios for the control? How does it link to the iron deficient plant? I think they've either worded this really badly or nobody's understanding. Because yeah, this guy said, I think it is the ratio of protein to chlorophyll. I'm baffled. I am utterly baffled. Nobody else seems to understand how to get the answer here. Let's have a look at what Google has to say. Okay, well, they not only got the wrong answer, but they did the ratio of protein in chlorophyll is 31-9, but the mark scheme says it's 36-1. Hang on, guys. I need you to see this. Look, this is someone did all this working out to get 31-9. That's not the right answer. How has someone done like? This guy's been a teacher at the University of Nigeria for 2 years. How has he got the wrong answer? Unless the mark scheme is wrong. That is utterly gong for Googled me. That is flimblegasted me. That is the one biology question that if I got in an exam, I would actually top myself. The figure below shows transmission electron. Oh god. Why are we on question C? Have I got like a really shit like paper here or some shit? Because these questions are getting more and more stupid. Contrast the structure of the two cells visible. What does that mean? Contrast the structure. I mean, I guess it means describe the differences of the two cells shown. But I don't even know what these cells are. I've definitely gotten a stupid ass thing. That looks like a bacteria, so I'm assuming you could talk about slime capsules and stuff. Oh yeah. A has no capsule, B does. A has a nucleus, B has free DNA. Oh yeah, okay. Alright, that was actually a very easy question, just disguised as a really stupid one. Bro, that was a 5-mark question. Bro, what are these biology papers? That is actually a really nice question. It's disguised as a goofy one. It makes you think, what the hell do I do? All you're meant to say is, oh, that's a bacteria. You don't even need these images. You can literally just say a bacteria has XYZ, prokaryote has an animal cell has ABC. That's an actual lovely question. Eukaryotic cells produce and release proteins. Outline the roles of organelles in the production, transport, and release. Okay, well that's also a very nice one. You could just talk about how DNA is the one that releases RNA, but I'm not sure if that will be required. You can then talk about how the RNA goes to the rough endoplasmic reticulum, which contains ribosomes. The ribosomes perform, synthesize the proteins. These proteins go to the Golgi body, where they are checked and modified. The vesicles and then tape them outside of the cell. I guess. Yeah. Okay. DNA in the nucleus is code for protein, so you didn't even need to talk about. Wow, you wouldn't have even needed to mention half the shit I did. And DNA in the nucleus is code for protein. That's basic. I didn't mention it because I didn't realize the mark scheme was like easy all of a sudden. Ribosomes / ARIA produce protein. Yeah, they synthesize it. Oh wow, you could have even mentioned the mitochondria producing ATP. Wow, that was a nice question. I might not have even gotten any marks for it, though. I might have gotten, like, 2 out of 4 for that, because I was too specific. The Golgi apparatus package and modify. Well, I put check and modify. You could have even said the carbohydrates are added to create a glycoprotein. The vesicles transport it. Wow, okay. So I guess in an exam, if it says don't include details of transcription, you don't need to mention anything about DNA, really. You just say the DNA codes for proteins. And you have to mention the nucleus, obviously, because it's an organelle. Ayo, why is there a coloured image? The image below is a transmission electron microscope image of a plant cell. Suggest why a nucleus is not visible. Huh. This is a what? Why are the 1 mark questions stumping me the most? Why a nucleus is not visible? Is it going to be something stupid like, oh, because it doesn't have one? What you actually say about, ready? Huh. It basically was, the nucleus is not stained, or this is only a section of the plant cell. This is only a section of a plant cell, so the nucleus might be in another part. Bruh. Name the organelles S and T. T is chloroplast, S is cytoplasm. What? Hang on a minute, vacuole? How zoomed in is this image? That literally is the cytoplasm, no? Okay, well apparently that whole empty space is a you just got vacuoled. You just got vacuoled, bruh. I just got vacuoled jump scared. I'm baffled by that, because you can literally see the cell membrane there. You can literally see the cell membrane here. Where would it be? Okay, well, I just got jump scared by a vacuole. Give one advantage of viewing a biological specimen with a transmission electron. Transmission has higher resolution. And that's all? Oh, that's literally 1 mark. Okay, yeah. Higher resolution, so you can view the internal structures. Okay. Whoa, what the hell is this question? This is like some math geometry question. Bro, get out of a biology paper. Calculate the number of cells in the upper surface of the leaf. Bruh. What? Give it in standard form? The cells on the diagram below are part of a continuous layer of cells. Would you do 150 times 6? I'm kind of baffled by that one. This again looks like it's like a geometry question merged with. The shaded area is 150 micrometers. Micrometers to, I can't recall. Micrometers to millimeters is times 1,000. Millimeters to centimeters is times... Hang on. That's literally times 10. How would you times by 100 million? Correct answer for any number divided by 150. That is the most baffling question. Why is this paper be the most baffling one ever? Oh my god, man. Oh, bro, there's more math questions. I can't take this anymore. That's a 1 mark question, and you have to like do a lot of conversion for that one. Oh, Jesus. I guess to get to centimeters to micrometers, you would divide by 10,000. To get from centimeters to nanometers, that would be 100. That'd be 10 million, I believe. Yeah. 1 centimeter is 10, 100,000, 10,000, 100,000, million. Yeah, okay, I'm right. God damn, bro, these questions are disgusting. Identify structures D and E. Bro, you've shown me like the most grainy image ever. How am I meant to identify what the structures are? E, I'm gonna assume is a starch. I don't know what you call it. It's a starch something. And all it says in the thing is starch. Okay. E is starch. D would be a thylakoid. Oh, it's grana. Grana which stacks onto thylakoids. Okay. The details shown in the diagram would not be seen using an optical microscope. Why? Because it shows, I guess, all the internal organelles, which are smaller than the wavelength of light, meaning an optical microscope could not see them. Light has a longer wavelength, so it has a lower resolution. I don't know if I would have even gotten a mark for that. That's so vague. Light has a longer wavelength than... I don't know, bro. These mark schemes are so cryptic, bro. Why are the mark schemes more difficult to figure out than the actual questions? Name an organelle found in both a chloroplast and a ribosomes? Yes, okay, but you would have had to say 70 yes. Scientists determine the... Oh, bro, stop with the math questions, bro. A scientist determined the volume of a plant cell and the volume of organelles it contained. Nah, skip. I'll do this one later. Skip. Another centrifugation question. Describe 3 properties of this solution. Ice cold buffer. Well, ice cold, buffer and solution. Oh, I guess, yeah, same water potential to prevent bursting of organelle, yeah. Okay, the questions seem to get very easy from now on. It seems like I just got shit RNG. That is just a mitochondria, and you have to draw it and label it for 4 marks. That one I've literally done in school. W is the membrane, which is made out of phospholipids. Murine is what comprises of the cell wall in a bacteria. Mitosis. No, not mitosis. Binary fission. Okay, well there you go, guys. I'm bloody tired now. I've just done maybe like half an hour of biology questions. Apologies if I didn't read your messages or anything. Bro, I swear down, bro, the biology mark schemes are actually worse than the questions. How is that possible? Bro, trying to decipher the mark schemes for these things is so difficult. But anyways, I'll be back whenever for another one. Oh, hello, if you're still watching, Reese. I don't know if you heard what I was trying to say before, but basically I can't read chat without having to like completely turn my thing around, so yeah, I am good, hope you are too. I'll catch you guys in a bit, but goddamn, bro, these questions, I'm not looking forward to the AS level, bro, I am not. God damn.